All right, welcome. This is the SFPDASC. We're an affiliate program of the West Coast chapter of Parenteraldrugassociation, Association, connecting people, science, and regulation. Today's topic is COVID-19 vaccine, call for volunteers. We all work on the vaccines and medicines, but can we step it up and volunteer to ensure that the vaccines are safe? Today, we have me, Nick Kapp, the academic advisor for the SFPDASC, and we have a special guest today, Myles, who happens to work for Genentech, but also is a volunteer and has received a shot for one of the studies. Here is a picture of Jane. She's taking her selfie, getting the shot in there. So Jane, if you can introduce yourself, tell you what you do, and then talk about what you did and what the experience was, that would be great.
My name is Myles, and I've worked pretty much a lifetime in designing and implementing clinical trials and then submitting the data for regulatory review in all sorts of different indications. I did work at Genentech Roche until about August last year, and as of this year, I'm actually working for Covance, global CRO, and I'm working on decentralizing the clinical trial model. That is how we introduce technology into trials in different ways to improve patient and site experience. I call myself a clinical trialist. I haven't worked at the site full time. I've always been in the design and execution phase. And a couple of years ago, I volunteered to participate in the Verily Project Baseline study. And I was really interested in their mission, but really I loved most the experience of learning by doing. And so when the call came for volunteers in 3 studies of 30,000 patients, it was a no-brainer to me. I got to do this.
So this was for the COVID-19. Was this the first study that you were involved in for a drug or a vaccine?
Yeah, my first interventional trial experience. I got curious first when I saw the Moderna data and first reached out to see if I could participate in that trial. So I went to clinicaltrials.gov, as you might do, and started searching for information on the vaccine trials. And it took a little bit of know-how, even though I know a lot about clinical trials. I found it. And then I found the research site that was closest to me, which is in Sacramento, actually. But there was a research site open in Texas that was actually screening patients for anyone in the country. So I called them, and I was really impressed because it was a Saturday morning. Actually, I wrote them an email from their website. They called me back within about 3 hours. Again, it's Saturday morning. They're in Texas. It's about 10 a.m. my time when they call, so I think that makes it noon their time. This was a study coordinator in their Austin site, and she was really triaging to see where might I fit. Was I a reasonable candidate, and was there a potential geolocation that matched me? She identified that I might fit with the Sacramento site, but the Sacramento site wasn't open. So then she took my name and put me on the list. Time goes by. About 2 weeks later, I see that Pfizer has completed their analysis and they're expanding to the phase 3 cohort of their vaccine trial. So I go back to clinicaltrials.gov search, find that the closest research site to me is in Walnut Creek, and call the Pfizer hotline for trial matching and say, listen, here's where I am, here's where I want to go. They do the pre-screening questionnaire. Great, I'm on the list. But nothing happened for more than a week. And at this point, I have a good friend who's in the trial as well, and she's in Boston. We're comparing notes, not about the experience, but just about the experience in the recruitment funnel. And it's not to lay blame. It's just like, this is puzzling. Both of us have been recruitment experts for years. What's going on? The way that I solved for this ultimately was by getting the number of the research coordinator at the site in Walnut Creek. It meant I kind of did a workaround to the recruitment company. I wasn't violating the parameters of the study, but I had to take enough action to find the individual who was attached to that research site and really express my interest. So I need to quantify this a little bit better because I'm going to give a talk about it in a couple of weeks at the DIA. I think it took between 5 and 7 tries on my part.
What's the DIA?
The Drug Information Association. Another great topic for your students, actually.
So being involved in the industry like you were able to get an individual's phone number and stuff, or no?
Yeah, I did that all through triage, like triangulating the information on clinicaltrials.gov, then going to the website for the research site that I thought they were talking about in Walnut Creek,
Okay.
and then eventually finding the name of an individual to call at that site. So I
Okay.
wasn't pulling any strings, but I was using what we'll call some in-the-know detective skills on how to triangulate information.
You feel really safe. So the mRNA, this is a pretty new vaccine, but reading the studies, you feel comfortable using the vaccine.
The study designs between these trials are actually really similar. Less Dr. Fauci and NIH for mandating that, because it means we're going to have an apples to apples comparison, even if the platform the vaccine is built on is different. So I've only had my first inoculation of either the placebo or the vaccine, because it's a 50-50 randomization. My second inoculation is on Monday. The first day took about 4 hours at the research site, and it was just because they were being super careful about patient safety, as well as making sure they were following all the protocol requirements. But great experience, fabulous people. Can't wait to go back. They called me this morning to remind me to be there at a certain time on Monday.
So they were taking your vials, the statistics, they were taking your history and kind of everything else that they're doing. And then they take a blood sample for a baseline?
They took all kinds of things. So there was a confirmation that I actually did meet all of the study parameters. I did complete the consent process using an electronic platform in the context of the clinic. And so I got to meet with the PI at the site who double verified everything. Then we did a number of physical assessments, vitals, etc. And they took a lot of blood samples. So yeah, there'll be some baseline samples for antibody levels and immunogenicity, but probably other things too, like basic liver function chem, CMC. CVC, sorry. And then I got to do an active COVID test. And before we did it, they told me, please don't expect to get the results from this test in any time that you need to make a decision, because they're going to get bundled with all the study participant results. And they told me how to manage the discomfort of the test. I had a million questions for them, Nick. I mean, probably the reason it was 4 hours long was because I was asking them a billion questions.
Yeah.
And then I got the shot, which was, by the way, interestingly, the study is unblinded to the person administering the inoculations.
Huh.
Yeah. And that it's being controlled very rigorously, but it's actually the best way for them to be able to move at speed, because it was going to be too time consuming to generate a placebo that looked exactly like the vaccine. So the needle is actually blinded to you as the subject with a label over anything that's in it. The person administering the inoculation knows what it is and is confirming it based on the randomization. Does that make sense?
So that ensures that you get the vaccine or the placebo, and then they're going to do this twice. So you need to be able to make sure that you get the vaccine or the placebo definitely that second time.
Yep.
But they did their best. I mean, you could, even if you've asked them, am I getting the vaccine or the placebo, they wouldn't have told you. So it's still blind to you.
That's right. Yeah. That's really important for the maintenance of the study and parameters. And I asked the coordinator at the front end, like, what's going on in terms of the adverse event profile from your patients so far? And she giggled a little because she said, well, there's clearly a placebo effect. And I said, well, tell me what you mean. And she said, we got a 50-50 split on who's getting what, but we got about an 80% report of symptoms, like injection site reaction or soreness of arm, the things that are typical with a vaccine, but nothing atypical. And you asked earlier about whether or not I thought this was safe. I did, because I looked at the data on immunogenicity and adverse event profiling from what they published from phase 1. And I actually saw a really great article just the day before I went for my inoculation that outlined how Pfizer had selected the candidate they took to phase 3 and why they selected it. And long story short, the selection was based on immunogenicity, but the mRNA strand there using actually codes for the entire spike protein. And they had considered using only a partial spike protein code, but they saw better results with the full protein. So that's what they went with. So they're being pretty transparent on their decision-making process and what they're selecting. So I felt pretty confident. And it's also shown really good immunogenicity in older age group populations with no significant adverse event profile.
No, it's good. That's good to hear. But again, that's a small study. That's why they're doing a large study with 30,000 to really see. And then they're going to follow you for roughly about 2 years. Is that correct?
Yeah, that's right. And as somebody who's working to decentralize trials, one of the things that I find really interesting is none of these visits are done remotely. So they're all in-person visits, which kind of makes sense in some ways, but the innovator in me is always like, well, how could you do that differently?
Yeah, so if you move across the country, I mean, in 2 years, it is fairly likely that you may move. Would they be able to take you at another trial site, or do you have to come back to the California site?
No, they could do that.
Okay.
In fact, I would probably figure out a way if they didn't know how. But that's pretty easy to do as long as your data can go with you.
Mm-hmm.
Yeah. And the first couple of visits are the more intense ones because that's where the inoculations happen. The follow-up visits are really just vitals and immunogenicity testing.
So they would take a sample of blood and see if you've developed the antibodies.
That's right, and if they persist. And I think that's going to be very interesting over time, right? Like my husband, Peter, is also in the trial. And let me tell you, his experience with his peer group is really different than in mine. Another story. But when you come home from your first study visit, you come home with a kit for a self-administered nasal swab in case you develop symptoms of COVID, because you will have to go for your regular treatment through your PCP, but they'll also want a sample to determine whether or not you have active virus based on your symptoms. And then you may be called in for additional blood work, right? Because ultimately the test of this is whether or not the immunogenicity persists and if it mounts a response to a threat from COVID.
Okay. So, so you didn't get any instruction that you're supposed to go about your life as you have. You don't know whether you're in the control group or the other one, but what they're looking for is a small percentage of people in their just active daily lives are going to get exposed to COVID and get the disease. And so the idea is that they look at both sets of individuals in the trial, and they're going to see percentage-wise, because not everyone was going to get it. No one's going to shoot you with COVID-19 to give it to you. They're not expecting you to go out and go to frat parties and stuff like that and get the disease. Just do what you do, and they want to look and see, does it protect you?
There were 2 things they requested, actually. They were not difficult, but they just were clear. Please don't leave the state for 2 weeks after your second inoculation. And don't schedule your flu shot until 3 or 4 weeks after your second inoculation. And that was because they don't want to get a covariate in the data on immunogenicity, but also, frankly, and I appreciated this, they said, we're not sure that the duration of the flu shot's viability will extend through the whole flu season. So it's kind of in your interest to have a later scheduled flu shot anyway. And Dr. Fauci said that yesterday on something I watched with him too. So it is to preserve the study, but it's actually probably going to work in my favor. Other than that, and so one interesting thing you just mentioned, I'm in it in part because I met the criteria and I'm in sort of the middle age group. but I have pretty low exposure risk. My husband is in a similar age group because they've got all kinds of cohorts and strata. But he's in the higher risk category for exposure, not because he's a healthcare worker, but because he's still running a lab and people are coming and going from the general public in the lab all the time, even though they're following social distancing and pretty good safety protocols, and it's a lab. A number of people come into the lab who've been exposed.
So you did this, we're in a very critical situation with the pandemic. And so it was your idea, I mean, kind of your job that you're doing, you're helping us, helping society anyways, but you felt that you wanted to be a part of the test for the vaccine to kind of do your part.
Absolutely. There's a volunteerism element to this. And there are a couple of, I'll call them, what's the right word? Self-interested reasons. One, that learn by doing experience is just golden for what I do in my job. But second self-interest is I want my son to be able to go out in the world again. And I want me to be able to go out in the world and get on a plane. So if I have a 50-50 shot of getting something that might be helpful, bonus.
Do you feel that they're going to rush this? And so there's a big push. Again, you've probably seen that the CDC has directed the states to get ready on November 1st. To kind of have everything all kind of set up to get the freezers or whatever that they need. Do you have confidence that the companies and everyone involved will only release it if the data looks right, if the science looks right?
I do. I have a tremendous amount of confidence in that. And I'm biased, okay? Because I've worked in the industry a long time, but I've had the good fortune of working for people who believe that science is our true north. Like, yes, we want things for the betterment of humanity, but science is the true north that we follow in order to achieve those goals. And I heard the chairman of Pfizer make those remarks the other day very clearly, like, we will not be rushed. The integrity of this study is really critical because it's a public trust issue, but it's also a public safety issue. So when I heard the call to action for states to get ready, I actually thought it was ludicrous. Because this is, let's say the Pfizer vaccine works, and let's say they have data early enough to make a decision on that, even before they get through 30,000 patients worth of endpoint data. Scaling up the manufacture of this is not something you do overnight. And then there would have to be, I think, a really clear triage process to determine who gets the vaccine and in what order. I would be at the low end of that list. So we might have a need to supply the vaccine to frontline healthcare workers or those who are in high-risk situations. But I don't think we're going to have a vaccine at scale by November, or actually by March, or maybe by June. So I think there's a little bit of patience that comes along with science.
No, I think that's definitely true. Is there anything else that you want to say?
I'm really excited for being in the trial and for being able to talk to other people about being in the trial. I truly consider it a privilege. But I'm also really excited, Nick, because this is a difference generationally in the public's awareness of medicines and how they're developed and the need for volunteerism. So part of my mission back in my real job is actually to capitalize on that new awareness, and most especially not to violate the public trust with that new understanding. because this has been the deal breaker for the ability to develop medicines quickly for years. I'm really thrilled because I heard that Pfizer is more than 2/3 finished recruiting the 30,000 patient trial. It opened July 23rd or something.
Yeah.
That's incredible to me.
Yeah. To me too. I mean, it's amazing. Yeah.
Moderna has slowed down enrollment because they're starting to fill all the slots they need in certain states, and they're looking for diversity in the population, which is also a good message, right? The AstraZeneca situation is interesting, and we'll see what that does, not just to the AZ candidate, but also to the willingness of people to participate in trials and then to accept whatever outcome happens.
Yeah, just for our audience, the AstraZeneca trial, their vaccine is based on the adenovirus. It's a different kind of a different modality for that, and then they had an adverse event. But you know, I'm really proud of it because they had an adverse event, and rather than bearing it and going through with it, they pull it out. They did exactly what their study said. If you had these events, you know, you need to shut it down and monitor the situation. And so I actually feel really, really good about that.
I do too. But you and I are looking at that through the frame of reference of understanding the scientific principles at play, right? But you're right. In a bad situation, a company could have chosen not to be transparent and not to take those actions.
Actually, I'd like to open it up if you're okay with some of our students asking questions. That would be great. Maria or Priscilla, do any of you have any questions?
I actually do. Last week, we were talking about who will be willing to participate in the study. I told them that I actually wouldn't. So my question is, what would you tell people like myself who wants to help but is scared of the risk?
You know, I don't think I would tell. I think I would ask. Like, I would want to explore with you what your concerns and worries were, because they're all valid. And some of them may or may not actually apply in this particular study. So, for example, Nick probably knows I did this because I put it on LinkedIn. By the way, the PI told me I was the first patient she'd ever seen take a selfie under their poster. Because I was on a mission to help them recruit. So the thing that I always want to do is try to work with particular concerns. And by the way, often they're from your family, not from the individual. So when I started examining this possibility, I started recruiting my husband. And he had very different questions than me. But on social media, what was fascinating to me was the number of people who actually work in the industry who expected that by participating, I was volunteering to be exposed to active COVID-19 virus. It's like, that is not the way that this study is designed. It is the way that a lab study might be designed, an in vitro study, but that is not an ethical way to conduct this trial, knowing the severity of disease and how infectious it is. So I think that was really fascinating because these are very well-educated people, some of whom are immunologists, and they're right. In a truly classical experiment, you would do that, but you can't do that in this setting. So we're doing a surrogate endpoint, which is immunogenicity, as opposed to the ability to fight the virus.
Basically, if you were in a lab and we were doing this with mice or goldfish, you would give them the vaccine and then you would do a direct challenge on that.
Yeah.
Just with human beings, and it's unethical to do that. So one of the reasons why we need to do 30,000 is you want to have a very large group, and because again, just living our lives, about 1% of us are going to get exposed to this, whatever it is in our area. And we want to see is the people who get the vaccine, hopefully none of them will get the disease. And then the normal 1% of the other population will get the disease.
that's going to take some time for us to prove, right? And so the other benefit of the immunogenicity surrogate marker or endpoint is we can assess this more quickly than waiting for the real world data to show up about exposure, right? Now they'll be tracking that, and over time that will be published as part of the study. I looked at it when I read the study parameters, but the immunogenicity is a viable way to assess the efficacy of the vaccine without waiting for 2 years. Lots of people have been worried about the placebo, like what's in the placebo? Could you get sick from the placebo itself? The placebo is saline in this case. It's not in every study, but in this case it is, and they told me straight up. Let's see, what else have people been worried about? Some people were actually worried that I was even going to a clinic, because wouldn't the people who are already sick be going to the clinic? I was like, well, maybe, but probably they would be going to their PCPs, primary care physicians, whereas this is actually a research clinic conducting many different types of studies, many of which are not at all related to COVID-19. They're doing diabetes studies and obesity studies and some oncology studies, but they're not actually doing any active treatment studies for COVID-19. They're doing a vaccine trial. Does that make sense?
So I actually applied for the AstraZeneca trial. And in some of their data, they said, if you want to do this, talk it over with your primary care physician and members of your family. Did you talk about this at all with your primary care physician, that you're going to do this?
I didn't. It would have been a good idea, but I felt informed enough that it was okay. By the way, I did sign a release to allow data exchange between my primary care physician and the research site if that's necessary. My husband, on the other hand, had some different concerns, and so he didn't consult his primary care physician, but he did think about it from some different angles based on his own history, especially with his reaction to the shingles vaccine. And so he asked a lot of questions about that when he was screened by the study coordinator. In fact, he thought that that meant that they were going to import all of his data from the electronic health record that he's generated over the years into the study data set. I was like, no, that's not how it works. But you see, all of these little tiny details kind of help me understand how complex these trials are and how many misconceptions there are out in the world.
Does anyone else have any questions?
Oh, I actually have a question. So you're obviously coming from a mindset that knows clinical studies as you actually, design them. What would you say are things that a normal or like a everyday human being would actually have to know before going into these type of clinical trials? What are some risk factors that they have to take into account? What are some just some different things that they should know about the trial before actually participating in it?
You know what? The coordinator was really good at explaining all of that. And you can tell she's really well trained and experienced. So the first thing was, do you really understand what we're asking of you? Do you understand that half of the people will get a vaccine and half of them won't? This is way before consent, right? This is just, here's what this is all about. Do you understand that this is, I think it's 6 on-site visits as well as a patient diary over the course of 2 years? And are you willing to really commit to coming to all of the visits? Because if you don't, we're not really going to get the data we need, and you're not going to get the oversight you need. So that was good. A lot of the risk factors were really centered through all of the pre-screening questions, Jonar. So she asked a lot of questions about my prior medical history, and that opens the door for a whole bunch of risk questions to come out, right? Like, did you have a blood transfusion in the last 28 days? Have you had a vaccine in the last 28 days? Have you received any medical treatment in the last 28 days? You see how she's going further and further down?
Yeah.
So she's assessing your risk, but it's also prompting you to think, well, what do I really need to ask? So I asked a bunch of questions about my prior medical history and whether or not that was a problem, not necessarily for me, although part of it was for me, but also was I not actually going to fit the parameters for the trial? I've dealt with lots of protocol violations, can you tell? But I also wanted to understand, is the mechanism of the virus likely to cause, the mechanism of the vaccine likely to cause any issues with my prior medical history? Because I have an autoimmune disease. So that was a really important thing for me to sort out. I was pretty confident, but I wanted to make sure, based on their knowledge, and also to make sure that autoimmunity issue wasn't gonna kind of skew the results I would provide.
Yeah. And I also have another question. You design these clinical trials also, and what would you say if, and you touched upon this actually, what would you say if there was a vaccine provided prior to the election day? Because a lot of these things can get really political, especially with, everything that's going on right now. Would you be confident in a vaccine that's created that fast, or?
That's not a yes or no answer. That's the I want more data answer.
Yeah.
I would want to see the data about the approval. I would want to look at the package insert. I would want to investigate the company that was manufacturing the vaccine. And as Nick said, understand what's the technology being used for this vaccine. Is this an attenuated virus? I'm not so sure I'm good with that in this case. Or is this mRNA, or is it an adenovirus? What exactly is the scientific basis of this immune response? I would want to understand a little bit about the parameters of the studies used to approve it. Like, if it's that fast, it might be completely valid, but I think it's back to that true north of science. What really concerns me is that just in the media at large, I've seen a high proportion of people indicating that they're not willing to use a vaccine even if one is approved. And that really worries me for a number of reasons, because I think we're going to be dealing with this for a very long time if we don't come up with a vaccine. I didn't live through polio, but when I look at what happened in that time, it feels really similar to what we're going through now in terms of the changes to public behavior and the fear that was present. And it took a while for that vaccine to be developed and then distributed. We have the benefit of speed and transparency and technology that didn't exist then, so it will go faster. But in my opinion, we really cannot allow politics to interfere with this. We can use the political engine to fund and to put rigor into the process, but not necessarily to relax.
Awesome. Thank you for answering all my questions.
Well, Jane, do you think, again, these are extraordinary times, and it's kind of really interesting to see that, if we can speed this process up and still maintain the safety that we have before, do you see this kind of changing, the pharmaceutical industry, to see that, drug approvals, vaccine approvals get out kind of much faster than they have in the past?
I think there's a real possibility of that for a couple of reasons. One, that public awareness has shifted. Two, I think we're seeing the dawn of what I'm going to call platform-based trials. And by that, I mean using that really consistent design. So you do an apples to apples comparison, or maybe you even evaluate multiple new molecular entities in the same trial. That would be a game changer, because then you don't need 30,000 patients for each trial. Maybe, I'm making this up, okay, I'm not a statistician. Maybe you need 50,000. but you know that you have enough control data and enough sample data from each of the candidates to make a viable choice. I think the regulators are also very open to shifting how technology enables speed. They still want rigor in the conduct of trials, and they want data comparability, but they're really open to receiving data in different formats than we've used in the past. And that, believe it or not, has a huge speed factor. Like I won't get into it, but if you could actually just submit the data from a patient's electronic medical record instead of through the entire case record process that we use in trials, would be an immense game changer in terms of speed and efficiency. The challenge is knowing that you're getting what I'll call congruent data, right? Like what came out of this record for this patient is the same field as for this patient in this record.
Well, Jane, thank you very much. Sorry to keep you here a little bit longer.
I love it. It's my passion. I guess you can tell, right? But if there's anything I can do to help you and your students better understand research processes, clinical trials, not just as a participant, but like how do you make these trade-offs when you're thinking about using technology, I'm open.
Okay.
That would be really great. Thank you so much.
You're very welcome. And thanks for the opportunity. I really appreciated it.
Okay.
It's our pleasure.
Okay,
Thank
have
you.
a great weekend. Stay
You
healthy.
too.
Thank you.
Bye,
Thank
everyone.
you. Bye-bye.
Bye.
Bye.
Thank you for tuning in. This is Kapp Blogs at SFPDASC.